Hi, everyone. It's Dr. Tim and Hillary for another Dr. Tim's Aquatics podcast. How you doing this morning, Hillary?
I am doing good. I'm very excited. Reefapalooza is this weekend, and I can't wait. I'm
Yes.
looking so forward to warm, hot, sunny weather, and Reefapalooza, of course, but warm, sunny
Uh,
weather.
uh, well, that's what happens when you move to the land of rainfall and green pine trees. So, all right. So today I thought we'd go down and tiptoe into a little bit of a rabbit hole or rabbit hole. So because I wanted to talk a little bit about the basics of aquarium microbiology. And I don't mean just nitrifiers, but just aquarium microbiology in general. Microbiogenomics is the up and coming hot word. And there's a lot of m- misinformation in there. Is that a good word? I'm being kind.
I
Uh,
think being kind is good because if you're not familiar on this topic, and I feel like a lot of people are not, then they don't know what's right and what's wrong. And so I feel like being kind is probably a good way to start out.
all right, I will be kind. So let's start with a big, 20,000 foot view. Well, let's define biome, microbiome, genomics. So a biome is basically, usually if you look it up on Google or someplace, it's going to say it's a large environment that encompasses everything. And they'll show, a coral reef is a biome, the water, the reef, the rock, the algae, the fish that swim in, you know, it, or a redwood forest would be another biome, or an Arctic tundra. They're very different. When I say Arctic tundra, you're picturing snow and just that whiteness. If I do redwood trees, they're just huge, tall redwood trees. In the middle of the day, you can be in a redwood tree forest, and it's like the eclipse from a few weeks ago. Coral reef, obviously, scuba diving under the water. So those are very different and they're very distinct. And now we go into microbiome, actually, let me come back a little bit. There's a lot of research being done on coral biomes and the coral holobiont. And this is a scientific study because the idea is that a coral by itself is made up of all these polyps. And so you've got the polyp, but inside the polyp, what do you have? You have the symbiotic algae that provide the nutrients that photosynthesize and provide nutrients to the coral. But then you have these corals that are years, decades, centuries old, and they grow together and they form this whole community. And so it's looking at not just the coral itself or the symbiotic organisms in the algae, but how the corals get along and then how fish and everything interacts. And then you can dive deeper into our aquarium, and I'll come back to the coral in just a minute. And you've got the aquarium. And more, everyone realizes that microorganisms are really key to having a nice aquarium. And when I say microorganisms, yeah, I'm talking bacteria, um, archaea, cyanobacteria, dinoflagellates, protists, all these small organisms. I'm not so much talking about the fish themselves. And the idea being that there is an assemblage of organisms, the microbiome, meaning, the bacteria, the microbiology that's involved in a coral reef that if you have, well, I mean, good aquarium, when I say good, I mean an aquarium that everybody would probably say, yeah, this is beautiful. It's not overrun with cyanobacteria or algae or dinoflagellates, but it looks really nice. That aquarium has a microbiome, an assemblage of all these bacteria and archaea and microorganisms that is completely different than the assemblage of a bad aquarium. Bad being, it could be that this is an aquarium that gets a lot of diseases, an aquarium that's overrun with cyanobacteria or dinoflagellates or bryopsis or something like that. There's not a lot of proof of that. There's not a lot of proof of much of what I'm going to talk about. These are hypotheses that have to be tested, and it's going to take a lot of time and a lot of testing. But then if you let's just go with that and agree that okay, good aquariums have a microbiome that's different than bad aquariums. And further, if we could identify those organisms in the biome of the good aquarium, that we could then grow those good bacteria. And instead of just seeding your tank with nitrifiers, you could seed your tank with the good microbiome and establish that good microbiome from basically the get-go and not have cynos and dinos and all these problems. So, Hillary, have I kind of set the stage there that makes sense?
I think so, but I do have a question what you just said. Like, if you set the stage for all of the good positive things, what happens when you add a new fish or a new coral or some other sort of cleanup crew invertebrate? Don't you open yourself to the possibility of bringing some stuff in? Or is the good microbiome that you've already set up strong enough to battle that out and not give it a fighting chance?
Well, those that is an unknown. Yeah, what if you establish the good biome, how does it maintain itself? Or does it become so strong and what does that mean? And those are all great questions and those are unknown questions. Um, because not only do you have to factor in you're going to add a fish or a coral or something like that to your tank, but you're feeding the tank. And what are the choices of food? Say you're feeding a live food, or even a frozen food. Frozen food doesn't mean the bacteria or viruses, we're not even talking about viruses, are dead. So there are inputs to the system all the time. And is the system like humans generally most people are healthy 99% or 99.99% of the time, and because there are the cold viruses everywhere, we're just coming out of winter and the flu season, COVID, all that type of stuff. Why aren't humans sick all the time? Because your body has an immune system that can generally fight that off unless you're weakened or something like that. So, can the biome fight off these things? And the answer to that is unknown. The what research we do have is basically the human gut biome, because there is a consortium, which is a group. Consortium is just a fancy word for a group. Um, but that means this group interacts of bacteria that live in your stomach. And when you get when that consortium is upset because you're taking antibiotics or you're drinking too much or something like that, and you start killing that off, you have an upset stomach that leads to to diarrhea and things like that because you've upset the gut biome, and you try to reestablish it with some of these yogurts. And like everything else, there's some good ones and there's some bad ones, and the research is to a certain point, but the marketing of these products is kind of past the research. But that's what everybody's looking for is can we provide at base, at the base level, some sort of group of microorganisms that not only keep, your gut healthy, but let's get back to the aquarium, that keep your aquarium healthy. And I mean, I think eventually, within some reason, we can. But I think um, there's just not enough research for that right now. So, right now, any claims to that, um, I don't think are true. I mean, you might think, well, Dr. Tim, you make your waste away bacteria, right? But remember, those bacteria, one, were isolated from aquariums. And two, we don't claim they're going to solve all your issues, because while there's a famous saying from an author, Stephen Jay Gould, that he was a paleontologist and dealt with um, dinosaurs and big ideas, and he wrote a monthly column for Natural History magazine that was really great, and unfortunately passed away at an early age. Um, he wrote about how we live in the age of bacteria, and bacteria have always and will always rule the world. And it's the same with your aquarium. The bacteria are key, but the issue is we change things. And this is, we add filters, all sorts of things that we do to remove bacteria from the water column. And if you listen to me at all, I talk about that, where we unintentionally manipulate the biome by removing bacteria from the water column, but we leave the nutrients, and those nutrients are their money, their fuel. They're going to be used by something, and that something is dinoflagellates, hair algae, cyanobacteria, something. It's not going to go to waste, especially when you warm the water up and put all these lighting systems that we have these days that are incredible. Um, it's just going to do something, and so how do we deal with that? And that's part of the biome, as we're looking over this biome, and we're, like you said, Hillary, we're adding food, we're adding organisms, we're changing water, we're doing this and we're doing that. How do we maintain some type of stability? Do we maintain some type of stability? And the answer is it's unknown. And if you say your tank is really bad and you had this magic elixir, does just adding it mean you're going to take over? That's unknown too. You generally, because you can think about it as invasive species. We nowadays we travel a lot, and there's all sorts of organisms, macroorganisms, microorganisms, that easily move from one part of the world to another where they're not uh, native. And while some do get established, most don't, because the community is occupying all the niches that are in there. Sometimes that doesn't happen, or there's a perturbation, something upsets the system, and a new species can be established. But that takes time, and it doesn't, while when it happens, generally the results are pretty catastrophic to nature. It's not happening every day, all the time, every place, if that makes sense.
Oh yeah, no, absolutely. It's interesting that you bring up invasive species because it's gardening season here and I'm purchasing seeds online to plant in my garden. And the thought has crossed my mind, like, are these native seeds to my area? Like, yes, they'll grow here, but should they be here? And I've actually had to think about that. And thinking about it in terms of our aquarium, like there's nothing in our aquariums that is probably native. I don't know. I know a few people, like there's a guy in Florida that's got these local tanks that totally make sense. But, a good majority, I feel like, of our fish and coral come from Indo-Pacific.
Yeah, I mean that's it. But people mix it all the time. I've talked about that. I when I do an aquarium, I like to keep an aquarium that's geographically related. Even freshwater, I don't like to mix, Southeast fish from South America with South American fish and things like that. But that's just my preference. I don't know how much we're going to be able to change. Pausing here, folks, because this is not a prepared speech. we talked about this before we got on, and I said I'd give this a shot. But if you think about it, like Hillary, you say you have a tank, and sometimes people have this that just always kind of doesn't look that great, no matter what you do. Because how do you change things? How do you change things in nature? Your gardening, um, Hillary, if you were to just go and turn the soil wherever your garden and plot is and not do anything, would you get what you consider weeds, or would you get something that's more native? And maybe they're native, we just call them weeds up there.
That's an excellent question that I was actually talking to my neighbor who is a biologist up here, and she was saying, if you turn up the soil at least in my area, it used to be a prairie. And so while you might get a bunch of non-native weeds at first that come and fill in like 2 or 3 years, you're going to have a whole bunch of like wildflower species and stuff that have been dormant and buried for a while that will eventually pop up if given the chance.
Right. And see, that is basic ecology, and you have what's called R species and K species. R species are the colonizers. They take advantage of a new niche, something you went up there and you turned it over, made all this soil and exposed it, and they rush in there, they grow fast, and they initially take over. But as your neighbor said, given time, they will be overtaken by the native species. And this is a concept that I find people in the aquarium industry don't really understand. When you look at what's called a climax ecological zone, a steady state, redwood trees, the grasslands of Middle America, the savannas of Africa, there's actually very little diversity. Diversity being defined as number of species. And what I'm finding in the aquarium industry is that people are, well, we need to be more diverse, or the system needs to be more diverse. Well, why? Who says that? Why does that? Because that's not if you're trying to get stability, and if you look at nature, the ecosystems or biomes that are stable don't really have a lot of diversity. And that's not what nature generally does. Is there isn't. And even some coral reefs, I mean, you've seen the huge fields, and it will be dominated by one or two species of corals. Now, a hurricane comes through or something happens and it mixes it up, and you get the colonizers, the R's, but eventually they get replaced by the K, the climax species that tend to take over. And that's just repeated over and over in nature. So this thinking that we need diversity for diversity's sake because it's going to lead to a more stable environment is actually not correct. Diversity means that the environment is not stable to us. And well, why do coral reefs have, they say they're some of the most diverse. Well, it depends upon the area, but and there's a different definition there. So it's maybe counterintuitive, but if you start looking and Google biomes and things like that, you're going to see they have less diversity than more diversity. And when we get into the microorganisms, we don't know what they do. One, so what does the microbiome analysis allow us to do? So this is where you take a sample and you can send it into a lab, and now it's become relatively affordable that lab can come back and it can analyze basically the DNA, and you can do it for, prokaryotes and eukaryotes, the bacteria, and then if there's disease organisms, don't really do viruses, but they're parasites, things like that. That's a different database. But see, that's the key, is that in order for the the computer program, to tell you this sequence of DNA is this organism, that organism has to be in the database. And we do not have a complete database. So when you do this, you're going to get from phylum, genus, species, family, phylum, genus, species. You may get a bunch of stuff back that says, well, it's a proteobacteria. Well, what does that mean? That doesn't really tell you, it's not telling you a lot. And so you're not going to get results back that say that with 100% certainty, these are the down to the species level, for one. So you're not going to get tons of resolution. It'll come back, maybe it'll just say, Vibrio which is basically it's in the family Vibrio. Okay, well, Vibrio is kind of like the common cold, which is not virus. Vibrio is not a virus, but it's all over. It's opportunistic. Just because you might have Vibrio, an identification of Vibrio, doesn't necessarily mean you have Vibrio that's taking over your aquarium. So you have to be careful there. So one, we don't have a complete database that can ID everything. Secondly, okay, these organisms are there. What are they doing? Are they harmful? Are they good? Are they just benign? Again, we don't know. We know if you get a hit and it's relative numbers. So say it's you've got 3% nitrifiers. Well, we do know because we've isolated nitrifiers, and there's a pretty good record and a lot of work on nitrifiers, that they're doing nitrification. That's what they're there for. But what about all these other organisms? What are they there for? What are they doing? We don't really know. And
Okay,
then,
I've got a question for you.
yeah, jump in.
If we're just talking about a single individual sample, not like multiple samples over time, would we be able to even tell that much, or just say, hey, this is a static event, versus if
Well,
you had multiple samples at the same place,
it's
same
worse.
tank?
It's worse than that.
Oh gosh.
Because I've been reading up on this, and there's a paper that they took samples from sharks, but they would take a sample from the snout of the shark. This is not the external. They take the snout of the shark, the middle of the body, and then the cloaca. That's where the animal defecates. This is one animal, same time, right? The biomes were completely different.
What?
Yeah, yeah. So
All right.
yeah.
Okay.
Well, it's
Keeps
yeah.
getting more and more
Well,
interesting.
well, and that's the issue, is that where now let's go back to our aquarium. We have the water, which we manipulate, for better or for worse. We manipulate it. There's just no doubt about it. So we're and then even if, say, we didn't manipulate it, you have the water in your aquarium. You have surfaces that the coral, the rock, there's maybe hopefully lots of water movement. We assume water movement is good, and that it is. People, we've shown that people know that you just need to have good water movement in the aquarium. But you've got surfaces that are exposed to the light, then you've got internal surfaces that don't get much light. Well, what about the substrate? What's happening in the substrate? You've got the top of your substrate, whatever that is, and that's well oxygenated. But as you go down into the substrate, what's happening? It's accumulating organics. Organics are food for other types of microorganisms. The redox potential is dropping, so the meaning the oxygen level is also dropping. And I would contend that the biome, microbiome at the surface of your substrate is completely different than the microbiome at the bottom of your substrate. So you've got the water biome, surface biome, substrate biome. Is one more important than the other? We don't know. You know, we don't have answers to any of this. And there's a study that was done on the Ocean Voyager aquarium. Yeah, that's the big giant aquarium at Georgia. And they looked at the biome for a year, sampling in the water. And they found that it changes. Now, this is supposedly a stable environment. They don't come in one day and go, well, we're going to change how we're filtering things, right? That's the same filter, and they don't add a lot of animals to that. There's not a lot of changes, but there were definite changes in the biome over the course of a year. Is it due to weather
Is this
pressures?
their big tank? Like the
This is the
one with the
big,
whale shark?
yes. Yeah.
Could it be people that are getting in?
Could be anything.
True. I'm
I
just thinking
mean,
like because they allow, I mean, divers are one thing, but I think that's the tank they allow the general public. You can buy passes and swim with the whale sharks and scuba dive and all that jazz.
um, I'm, I don't know.
I was
The
thinking like when we were at the aquarium, like the dive staff all knew what, like, okay, before you get in the tank, shower, you don't want to have any lotion on, but there were times when we'd have to tell the new mermaids that were hired, hey, we know you want to put all this makeup and smelly lotion and glittery stuff on, but refrain from it because that actually messes up the tank. So
right.
it just makes me wonder.
Well, I'm sure it is. I just like uh, in more and more places, they don't allow you to have certain suntan lotions on because of the chemicals that are in them that affect the reefs. These things last forever. So the point of this is if you wanted to have, aquarium microbiome, the subtitle is we don't know anything. And that's not a knock or a negative. It's reality and it's an opportunity. And that's where, if you're listening to this and you're thinking, how could I combine my love with aquariums with my love for science and, really try something? That's an open field right there. There's a ton of things, and there is work being done. The Shed Aquarium has a coral biome project going on. The Georgia Aquarium has an active biome study group. There's aquariums in Japan that are doing this. So more and more people and researchers are looking at this. My only caution is that the aquarium business, and yes, full disclosure, I'm in the aquarium business, but I'm also in the aquarium research field. But the business part of it, as businesses tend to do, kind of tend to outrun the science. And right now, let's go back to the biome. You've identified it. You've done lots of work, and there's a pretty good certainty, that this is the biome. You figured it out Um, and these are the organisms that if you put them in your tank, you're going to have a really good chance of success, with success being a nice looking tank, and you're not going to have the cyno and the dino and things like that. Well, we have a big problem, Hillary. We can't grow these organisms.
We can't grow them yet.
We can't get exact. Well, we can't grow them yet, and we don't know when that's going to get solved. We can only grow 3 to 5% of the microorganisms that we can sample out of an environment, and an aquarium included. We don't know what nutrients, what micronutrients, what lighting. We just don't know yet, and that's a lot of work. It's kind of like another controversial field is trace elements in an aquarium. Which ones do you need? How often do you need them? What concentrations? Things like that. We don't know that. Um, and we definitely don't know that when it comes to the microbiome, and we don't know how to grow them at all. So, you know,
Okay,
go ahead.
oh, sorry.
No.
You said we can only grow 3 to 4% of what we sample. That still seems like a fairly large number. I mean, I'm assuming that when you do sample, like you can find thousands of individuals.
Yeah. Well, the limited sampling that I've done and the biome work is there's several hundred to a couple of thousand. It just depends upon what percentage. Usually there's, you know, then again, folks, this is limited, but there's a couple of hundred that make up 99% of the diversity that's out there.
Makes sense.
We a lot of times it just comes back not even at the genus level, just maybe at the family level. That it's part of this, but we don't really know what it does. We don't really know how important it is. Um, and then say you have this biome, you can grow this biome. Well, what about, like you just said, people, you're putting your hands in there, you're feeding it and you change the food, you add a new fish. What happens with all that? And and we just don't know. And it's complex, but that doesn't mean that it can't be figured out. It's just that you need to be asking yourself, these questions, and they're nice research questions for sure.
I agree. All right, I want to take a step back. So, probably like maybe 15 minutes ago, you were talking about diversity and about how more diversity isn't necessarily a good thing, that it tends towards less diversity.
Well, I don't know if it's a good thing. It's just if
Uh,
you look at good or bad, it's just that when you look at nature, the what we call climax communities don't have large amounts of diversity. Whether that's good or bad, it's just the way it is. So
okay.
so let's not put a good or bad label on it. It's just, pick any community you want to talk about, and it's going to have less diversity rather than more diversity. When it's that, when you name it coral reef or Arctic tundra or uh, Midwest savanna grasslands or something like that.
Okay. So I guess my question was based actually on some of the questions that we get asked from people. They'll talk about how they've used multiple products. They'll be asking us if it's okay for them to use XYZ bacteria product with our bacteria product. Say, for example, that somebody has used our products their entire life, and for whatever reason, they can't get a hold of something, and they try to add somebody else's live bacteria to their tank. Will those new bacteria even make a difference, or will they just get wiped out by the existing? Dr. Tibbs bacterias that were in their tank?
Well, it kind of depends in terms of uh if there's an issue. And the reason I say that is that if the Dr. Tibbs bacteria are occupying all the niches or are consuming all the resources, whether that's nitrate, phosphate, organics, or whatever, then adding more bacteria is not going to do anything. I always say it's a numbers game. And what's the first thing we talk about when people want to get rid of dinos or cyanobacteria is we try to change the numbers by going in there and physically removing. So you're a perturbation. You're the hurricane that's gone through the Keys and upset the coral reef. By going in there, your aquarium, and you're trying to get rid of as much of that cyno or dino or hair, whatever you're trying to get rid of, you're physically removing it from the environment and opening up those niches and resources, and then adding our bacteria to try to get them reestablished in there. Or if you have dinos, what do you've been to the shows? I'm sure you've asked this question. People come up and, I've got dinoflagellates. Well, what are your nitrates and phosphates? And invariably, not 100%, but over 90% of the time, the answer is they're great. They're zero. And so then adding anybody's bacterial product isn't going to work because the bacteria need some phosphate and they need some nitrate. And if you don't have any in the aquarium, they've got nothing to grow on. That's why the first thing has to be you've got to get your nitrates and phosphates up, because whatever you've done to the system, the water quality is determining what can grow. And right now, bacteria that might be helpful, cleaning surfaces, getting rid of organics, can't. And you've taken the tank chemically into a zone where the dinoflagellates, which are photosynthetic, so they can get their energy there, they can get assimilate nutrients from organic material directly, or the cyanobacteria can get nitrogen, the nitrate they need right from the nitrogen in the air, which the water is full of nitrogen because it's full of air. Um, they are able to exploit the chemical environment that does not allow the beneficial bacteria to grow, so that doesn't become established. So if there's nothing wrong in the tank and you just add a bacteria product, it's probably not going to become established. There needs to be there needs to be some resources that aren't being utilized by something else, or else you need to disturb the whole system to try to give what you're adding the chance to use those resources, and not the cynos and the dinos and the nuisance organisms the ability to use those resources. Does that make sense?
Yeah, definitely.
Yeah. But just adding, it's like adding fuel. So people add carbon dose. And the idea behind carbon dosing is that you're missing a chemical. You've got nitrates, you've got phosphate, but you need carbon. Every living organism needs C, carbon, and nitrogen, P, phosphorus. Aquarium's got phosphorus, it's got nitrogen, and the idea is that it doesn't have the carbon the bacteria need. But I'm sure you've heard this because I hear it all the time, and we'll probably hear it this week. And people have cyanobacteria. And what did they do? They added a liquid carbon, whatever it's going to be, vodka or some other product. And all it did was make my cyanobacteria, worse. Well, yeah, because the cyanobacteria are outnumbering everybody, and they'll take that carbon And since there's so many of them, all you did was feed the cyno. You have to disturb the system and get rid of that cyno to try to. The idea behind the carbon dosing is to grow pelagic or bacteria that are in the water column that can outcompete the bacteria or nuisance organisms that are living on the surfaces. But if you've got, ozone's making a comeback and UV and a roller mat filter and a micron's uh, sock, that is all removing bacteria from the water column and or killing the bacteria in the water column. But none of those things affect anything that's living on the surface. So you can try to manipulate the environment as much as you want, but as soon as the bacterium, splits into two bacteria, you've removed it or killed it by all those things that you have in the water column by making the water too clean, which is why, I kind of preach turn that skimmer off for 4 or 5 hours. Give the bacteria in the water column a chance.
Yep.
So, um, but getting back to the biome, I mean, it's an exciting field. There's a ton, I mean, almost all the questions are basically unanswered. And, it's going to be exciting to see. But at this point, just think when somebody's talking to you about that, just think about a few things that we know very little, and we don't know what organisms are the most important. We don't know how to manipulate and change. And do we want, it depends, we want change, because I've had this, I'm sure, Hillary, you've had this, and many people have, is that first good 6 months to a year that you set up a system, it goes through a lot of changes, the ugly stage, this stage, that stage. But then all of a sudden, and it's not really all of a sudden, but then something happens and the water has a clarity and a brilliance to it, and the system is just running. You don't have to interact, you don't have issues. It's almost self-contained, and the less you do, the better it is, and everything looks really good. And you just keep your hands off, right? And it just runs to a certain extent.
Yes.
Yeah.
Yes. It is glorious when you hit that point.
Right. And it's just, what is that? That now, and that's where it'd be fun to sample through that. But at that point, it's like that is a stable environment. And I would contend that the biome at that point, I mean, I would say that you're going to have much less diversity than when you began, because things that have become established and they're working together as the whole system, the whole little community. And the more hands off, the more you're not disturbing it, and it just runs and works. And that's the kind of the nirvana moment where it's just gorgeous. And you can tell with the water, because I've had this, and the water is just looks so clear, and the corals and the animals, and that's what you keep on coming back for. That perfect golf shot or that perfect aquarium look. And it doesn't happen overnight, but it does happen. And I'm sure underlying that is you're going to find that there's a microbiome there that is maybe not what we think, which is to be discovered. Be fun.
Yes. I think that's a perfect note to end on.
Yeah. So if you're going to be at Reefapalooza or on the responses, we can definitely continue this talk. It's to be continued in a whole growing field of research. So lots of fun stuff. So,
Absolutely.
all right, everyone. This has been Dr. Tim and Hillary talking about the aquarium microbiome. Please comment, join us, see us this weekend at the show, or we've got it's show season starting. We got lots of shows in the next 6 to 7 months. So come out, talk to us. We love meeting with people, right? So,
Yes.
all right. Take care, everyone.